So, first of all, I want to thank the organizers for bringing together this amazing group of people and this amazing school. Dr. Weil and Dr. Mohammed, Salam for doing this and for honoring me with this invitation. Now, I have a rather lengthy technical talk because it is a school where I want to introduce methods to study ion channels. So please, if I'm exceedingly boring, let me know, and then I will try to jump over a few sections, otherwise it will be endless. So, how do I do this? Okay. How do I advance? Okay. What do you do? Okay. Good. So I'll talk a little bit about the brain and how it works, and then ion channels, and in particular I want to focus on patch clamp methods where you actually see how an ion channel is working. So, if we reduce ourselves to a little model animal, then we need some sensory organs that detect the stimulus from the outside. We need a decision-making organ that somehow tells our effective organs, the muscles, what to do. If we see something, do we decide to eat it, or do we run away from it because it's likely to eat us? And these different parts need to communicate. And this part of communication is the essential, or one of the essential parts of neuroscience, of course, and of working of our brain. Now, because we have an eyebrow school here, International Brain Research Organization, I should briefly mention the brain, although I will very quickly go into very fine detail of it. So this is what we have between our ears. It is a highly specialized decision-making unit. And I was quite happy when we had this talk about the gut and its bacteria. It is not known actually that, or not known to so many people, that we have the same number of neurons in our gastrointestinal system as we have in our central nervous system. So, and also unfortunately, these two disciplines rarely interact with each other. When you talk about ion channels and neuronal signaling, you think brain and central nervous system, peripheral nervous system. You do not think gut. Although actually it's, many of the neurotransmitters are the same, serotonin for one, so there is actually a lot of crosstalk. So anyway, the brain is the major decision-making unit. I don't want to belabor this for too long. And now central nervous system, this has developed from sort of a network that spans the entire animal, as you can see in some leeches or starfish, hydra or grasshoppers. With the exception of grasshoppers, these are interesting because they can actually regenerate. You cut off part of their body and nervous system, it can regrow, which is actually an interesting part. And you can then see that in particular the forebrain has developed more and more. And the actual message from this slide is that we have in our central nervous system about 10 to 11 neurons with about 10 to the power of 15 synapses. And if we assume that each neuron can only be in two states, then we would have in our brain 2 to the power of 10 to the power of 11 possible switching states. And that is, as Carl Sagan, the cosmologist, said, these are more states that we have elementary particles in the universe. We have an extremely diverse central processing unit, and we are trying to understand how this is working. So, the brain is decision-making, it receives information from the surrounding, it passes it on and commands reactions to that. Some of them we know consciously, some of them are in the vegetative system, more or less unconscious, and we can try to control them, but we have lost this ability. Conscious and autonomous perception, and you can actually see this. I don't know if you can read this cartoon. In brain surgery, he says, this was a good one, poke his brain just where my finger is, and you can induce motion without actually the subject itself in control of it. So, this is now the simple animal, and now I want to take you into more detail of how communication between neurons and also between effector organs actually happens. And the heart of this is always, and you know this, that signals travel inside the nervous cell along an axon as an electrical disturbance, an electrical signal. And at the contact surface, we convert that into a chemical signal. So some chemical compound, a neurotransmitter is released and triggers a new action potential in the second postsynaptic cell. So again, this is how we can actually transmit information. Our neuron receives information on one end, it travels along, it can be, the longest neurons we have are about the length of our leg. And then here there are a new synapse where the information is passed on to the next cell. Very briefly, what are the chemical signaling devices that we have? These are neurotransmitters, and they have a refreshing property to them. They are very simple. We actually spend, although there is quite a few, their synthesis is not one of the most complicated stories in biochemistry. There are biogenic amines, amino acids, peptides, and some others. Most notable fairly recent discovery was nitric oxide as a neurotransmitter. We have heard about acetylcholine and, of course, particularly acetylcholine esterase, that makes sure that it doesn't hang around for too long. Monoamines like serotonin, catecholamines, dopamine, and so forth. Those highlighted in yellow directly trigger an ion channel. The others work through indirect signaling. Just give you a brief example how they are actually where we get them and how they are biosynthesized. Some of them are specifically made in neurons. Acetylcholine, gamma-aminobutyric acid, GABA receptors, we have serotonin, dopamine, noradrenaline. Glycine and glutamate are amino acids. We use them in all cells for biosynthesis and for many other purposes. They are just specifically packed away and stored for neurotransmission. It's just you need to store them in the right vesicles so you can use them. The reactants are in general metabolism. There's not so much special work on that. There are a few enzymatic steps. Mainly the synthesis is cytosolic. The only significant transporting process is the platinum vesicles. This is really the one simple story. I just have a few examples. And the bottom line is here, CoA, acetyl-CoA, and choline are simple chemicals that make acetylcholine in a single step. Glutamate can be decarboxylated with gamma-aminobutyric acid. Serotonin is made from tryptophan in 2 steps. Glycine is actually sort of interconverted from serine in a single step hydroxymethyl transferase. So, not so many enzymatic steps, no long pathways. We have them and we just utilize them in the right place. Ion channels, on the other hand, is a much more complicated story because ion channels are everywhere in our body. When cells communicate, they use some form of ion channels. So I have now expanded this signaling picture, electric to chemical to electric, a little bit. So this is what actually happens at the synapse. Arrival of the electrical signal triggers the release of neurotransmitter in the cleft between the 2 cells. This has been the subject of a recent Nobel Prize to Reinhard, I should have gotten it also, but not Tom Jake, the other one. Anyway, the name will come back to me in Texas. This is an own story by itself. We're looking at the postsynaptic part. We have transmembrane ion channels that bind, the cartoon is sort of supposed to have a sequence that bind an activating ligand and then a transmembrane ion channel opens. That activating ligand could also be an electric charge, a voltage change that moves and opens the channel. We'll look a little more into this. So, these ion channels are not just permanently open pores. They have some sort of a gate. So there is something, you can take it as a hole in the membrane, but that is guarded with a gate. There is something is needed to open that gate and to allow an ion flux across the cellular membrane. And studying the sort of ability of this channel to allow ions in and out, and this gate, this is actually the heart of what we do in neuronal signaling. So, a typical ion channel, therefore, passes through the membrane. It's a transmembrane unit, usually a classical about 20 amino acid helices that span the membrane. It has a water-filled vestibule, and somehow here there must be a constriction that doesn't always allow ions to pass, otherwise we just have concrete. And then, of course, water would not normally go through this bilayer unless we have this water-filled core where ions can easily pass. So, the structure of these ion channels actually follows a few simple principles. This is a potassium channel. It is voltage gated. It is classically made of 7 membrane-associated units. One of them gives a dip. This is a nice structural motif that makes the ion channel pore. We take 4 of those and make a tetrameric complex. You can see here in yellow the lining of the pore, and you can see sort of a positive charge here. And if you change the voltage across the membrane, this positive charge moves and pulls the channel open. And if you change back, or you have another gate that closes, then the channel will close again. The same principle is used in sodium and potassium channels. Only here you take those 4 subunits and you link them together. So this is one big protein that in the end makes the same class of voltage-gated ion channel. Sodium channels are sort of the same practically where you go. If you take a sodium channel of a frog or another animal or of a human or whatever, they all have the same characteristics. They open for a couple of milliseconds and close again. Potassium channels are the modulators. Actually, there is a huge variety of them. They're all different. So here there is much more variability already if you take different subunits that you can combine. Then we have ligand-gated ion channels. This is the part that actually I will talk tomorrow in the research presentation. They have a different transmembrane structure, one class of them. They used to be called the acetylcholine receptor superfamily. These days we call them the cysteine loop receptor superfamily for a certain cysteine loop in one of the domains. And they are made up of 5 subunits that go together to form a transmembrane arrangement. You can again see there is the same part of the protein that lines the ion channel core. And then there is a family of glutamate receptors, and if you remember the voltage-gated channels, here is the same principle of the pore. So they actually, glutamate receptors have a relationship to voltage-gated ion channels, but somehow they eventually turn into being activated not by a voltage pulse, but by a lipid that binds. And there is still a class of glutamate receptors that have a much stronger voltage dependence than others. So, these days, sort of the question is out, do we take 4 or 5 subunits? People lean to the fact that we actually need 4 subunits, again, a relation to the voltage-gated channels, and this is the family of glutamate receptors. There is actually, they're in our brain, so we do actually most of our thinking with glutamate receptors. When we calm ourselves down, it happens in the brain with gamma aminobutyric acid, GABA receptors. Benzodiazepines, Valium, activate GABA and make us drowsy. We move with acetylcholine receptors, and we stop moving with glycine receptors. So we distribute the tasks a little bit. And then there are other signaling devices that I just mentioned briefly between cells. Connexons, the so-called gap junctions, these are hexameric units now. They are direct electric short circuit almost between 2 cells. And we have a huge field of metabotropic receptors that actually are in our cells: muscarinic acetylcholine receptors, metabotropic glutamate receptors, serotonin receptors. Most of them, there are 7 families of serotonin receptors. One is ligand-gated, the others are indirect. They are activated with the ligand, and then either they open an ion channel, or they activate a G protein coupled receptor that then triggers intracellular signaling cascades. So, this is sort of the instrumentarium with which we do ion channel signaling. Now, how do we actually study them? How do we find that out? Again, here is the principles of these helix bonds that go through the membrane. Tetrameric, pentameric, and hexameric receptors is what we have with ion channels. This we know now, and it is quite an interesting fact actually. Before I go into that, with all ion channels, we know a lot about their function, and we know much less. This has only changed recently, again at the expense of a Nobel Prize, to people who found that out. We know much less, or knew much less about the structure. We know how this works with electric signal transmission in our nervous system since the 1930s, and we actually understand the structure since the 2000s. So between there we could look at the function, not knowing what it is. This is always the problem when you do protein biochemistry. With enzymes, it's the same thing. 1890, Emil Fischer had the lock and key model, so we knew how enzymes actually work. Their structures we began to solve in the 1950s with myoglobin. Until the 70s, 80s, we could work that out. And only in 1926, we knew that actually proteins were enzymes. So, anyway. I just give you now an overview. To the students, I would say don't memorize this, but just take the variety in. What kind of ion channels do we have? Voltage gated. We have sodium channels, potassium channels in all flavors, calcium channels, chloride channels, and another class of calcium channels in the rectifiers that are actually having multiple signaling functions even outside neuronal signaling. We have extracellular ligand gated. These I just looked at the classical glutamate and cysteine loop receptors. Nicotinic acetylcholine, glutamate, serotonin, GABA, P2X, ATP gated, and glycine receptors. We have intracellular ligand gated channels. Leukotriene receptors, IP3 gated channels, iodine receptors. Many of them deal with intracellular calcium concentration because in the cells calcium is a signaling molecule, not an ion. So there's a number of intracellular ligand-gated ion channels. And we have others. Mechanosensitive channels, mitochondrial channels, aquaporins, etc. Each of these channels comes in different subunits and different flavors. We have quite an array of different ion channels that we use for different purposes. That is a complicated set of proteins. So, now this is the interesting part. Ion channels are unique signaling devices. They don't bind something like an antibody or make a structure like keratin or collagen in our body. They are defined by a transient change. So this means that a transmembrane channel opens briefly and then it has to close again. And in this time it allows an ion flux across the channel. So we can actually, some people have likened an ion channel to an enzyme. And saying it's basically an enzyme that catalyzes ion flux across a membrane. We know the methods to study them, the electrophysiological methods, since the 1930s, and we're still a lot better about the function and the pharmacology, how we can actually modify their function, than we are about the structure of all these ion channels. So, again, the idea is that ion channels are a defined switching device. So, they need to somehow be activated, either by binding a chemical ligand or an electrical signal. Then they have this key process, gating. The channel opens. This is the signal that we measure. And then eventually the channels can deactivate a process known as desensitization. In the presence of the signal, they switch off again. And the key part is this gating process. Once activated, the channel goes between a closed and an open state. And if I can manage all, we can see at the end how these ability of the channel will go into the open state or not actually make the signal. And you can think normally a healthy channel has 90% of its time after activation in the open state and only closes briefly. And if it's diseased and mutation makes it close much more, and only open briefly, and then we have a problem in our signal. And these particular processes, how tightly does it bind the activating signal? How does it react? And how likely is it to open? How long does it stay in the active state? That is something that we can regulate with different subunits and other regulatory processes. I'm just mentioning phosphorylation as one of them. So, the methods to study. We can look at biochemistry, binding. If we enrich a protein enough, we can do binding studies. I'll talk a moment about that later on. We can try to isolate and purify the channels. Usually this is unhappy work with membrane proteins. We can try to reconstitute it. That is actually a more helpful method. Molecular biology, we can sequence them and clone them. Today with bioinformatics, we have the chance to compare sequences, and very quickly we know something about a protein which we have never seen or held in our hand. We just looked at the gene sequence. And we have methods of structural biology, which is difficult in membrane proteins. Anyway, in particular with something so variable